In 2022, a farmer in Michigan lost his 100-year-old cattle farm to the EPA due to chemical contamination, a terrible loss that left him in near bankruptcy. He isn't the only one. Farmers in Maine, West Virginia, and across the U.S. have been impacted by the same chemical contaminant. In 1981, a mother who worked in 3M's chemical manufacturing floor gave birth to a son with birth defects, requiring hundreds of surgeries to help correct them. A local investigation into the problem, done through drinking and groundwater surveys, revealed a disturbing amount of an insidious chemical everywhere that was sampled. All of these events trace back to a class of chemicals known as PFAS, or more commonly known as forever chemicals. This family of chemicals were mass-produced and dumped into the water since 1961 and have permeated everywhere. However, research into the impacts of PFAS is still new, and there are still new discoveries about the seemingly endless impacts that PFAS has on humans. Sadly, governmental regulations are also not able to keep up with the gray research that companies conduct to create new types of PFAS. Despite the prevalence of PFAS and the research being conducted, current testing efforts fall short. One of the only secure ways of PFAS detection is mass spectroscopy, a process available to universities and research labs. Individuals and companies have to ship samples to specialized labs for testing, driving up costs and hampering speed.
In order to combat this explosion in PFAS contamination, we designed a bacteria that will glow in the presence of PFAS, acting as a living sensor that works overnight without the need to ship samples elsewhere and wait for results. To engineer bacteria, we designed a DNA sequence containing our gene circuit that can be inserted into the bacteria. When exposed to PFAS, the bacteria creates a large amount of a signaling molecule. These molecules bind with a receptor protein, and together they trigger the production of a fluorescent protein called GFP. The signaling molecule should increase the amount of GFP produced per molecule of PFAS while also signaling nearby cells, amplifying the signal across millions of cells and increasing the sensitivity of the circuit. We then simulated the kinetics of our gene circuit in response to PFAS. In a cell simulator called vCell, we designed all of the components of our gene circuit and found that our bacteria would produce bursts of fluorescence in the presence of PFAS, giving us the confidence to move forward with the project.
We've also been working hard in the lab to start creating these mutant bacteria. With the help of genetics researchers at the University of Louisville, we have started to assemble the parts of our gene circuit through mini-prepping and ligation protocols. Our biosensor comes in kits that include the bacteria, which will be frozen during transit, swabs, nutrients to grow the bacteria, extra vials for testing multiple samples of water, pipettes, bags to store used materials which can be sent back to us for safe disposal, and an LED to make the bacteria glow brighter. The bacteria are an essential part of our innovation, containing the gene circuit necessary to detect PFAS. They will be provided on a frozen agar plate. The consumer will also receive a swab, which they will use to pick the bacteria off the agar plate when it is thawed. These bacteria can be placed into one of the vials provided, along with the nutrients that we provide that will allow the bacteria to grow. Next, the consumer needs to get a sample of water and use a pipette to transfer a part of the sample to the vial. With the bacteria, nutrients, and water in the vial, the consumer is all set and will be able to view the results soon with a fluorescent light that helps amplify the green coloring. We will also include an instruction manual to make it easy to use for people of all backgrounds. To ensure safety, our bacteria will be mutated to depend on a nutrient found in the liquid broth provided, which means they will die if released into the environment. Additionally, and most importantly, we are using a strain of bacteria that does not harm humans.
Shown on screen is our revenue model representing our major costs in addition to our major prices. Now, our kit will be priced at $60 because it doesn't use any high-tech or fancy gadgets, and also because of the low production costs of our bacteria, since we can produce large amounts of our bacteria and then parcel them out for distribution in the future. In addition to this, our two main costs will likely be the packaging and the distribution. One, the packaging, because we want to make sure that our product is still alive by the time it reaches the consumer. And two, the distribution, because rural centers will likely want to buy kits in bulk so that more field testers can validate their results with our kit. And this will allow our kit to be validated by even more governments and, in the future, even more companies. Because of our work with this gene circuit, we've expanded our network to include various faculty and adjunct faculty at the University of Louisville, and this will allow us to increase our propensity to innovate, which will lead to further innovations. In fact, most of our profits will come back into our company in the form of investment into R&D, so we can make sure that our PFAS kit is number one on the market compared to competitors. We are FluoroSense, and we are glowing the extra mile for cleaner water, and we hope you join our journey as well. Thank you.